Of the lecture after the break. See. Can you see my presentation now?
Yeah, I suppose we are all gathered. You can
Great.
continue.
Great. So we were discussing the patterns of ecological communities, or the space or the abiotic part of this environment in space and time. And regarding this community part, especially as a measure of it in terms of diversity, we see huge patterns across time. For example, if you visit a similar amount of forest in the tropical regions, you can easily see that 40 to 100 tree species per hectare. But you visit a similar patch of forest in the temperate regions, you only see 5 to 20 tree species per hectare. So why? That is the big question. And we only know this knowledge with quantified field methodology. Because if you walk in a forest, it is all green trees, that's just good. But we know there is so much difference in terms of the diversity, richness, and all the population rates of these two environments is because standardized forest ecology research. Lots of the ecologists, they established this forest plots, 50 hectare forest plots, and they did quantified science in it in terms of the amount of species, their identities, their growth rates. And all with this research, we now know that there is huge difference in these parameters between tropical and the temperate forests. And when we start to stack up all these information across the globe, we see other patterns. For example, one of the very famous pop patterns is the number of species changing from North Pole to the South Pole. And we see that the numbers of species have a peak around the middle of the Earth. So less species on the high latitudes and lots of species in the mid latitudes. So what can be the reasons? Of course, we can easily speculate. For example, we can check the resources. Of course, it's difficult to quantify the resources. Every species is using another resource. If you are talking about lions, they are eating gazella in some certain place and eating other things in other places. If you are talking about fox, it is eating mice. If you are talking about a bat, it is eating insects. If you are talking about a plant, it is feeding over the inorganic nutrients in the soil or in the aquatic environment. So it's a different, resource is different for each individual. And this is only the trophic interaction that I counted, who eats what. But resource is manifold. Availability of nesting space, availability of nesting material, and so on and so forth. All these things are resources. So it's difficult to distinguish resource. However, sometimes we tend to use a composite measure. And precipitation can be seen as a surrogate of resource because it brings, nutrient is a big resource, water is a big resource. And when you have high precipitation, you have more erosion, you have more transport of nutrients from one system to another, from atmosphere to land, from the catchments to the recipient environment. So you can imagine precipitation could be a good surrogate for resource. And when you plot annual precipitation across the observations across the globe, and then the number of species, you see that's true. When you have more precipitation, you tend to have more species. But is it the only pattern? What about the elevation? When there are more mountainous areas, high altitude systems, is it affecting? Yes, it's affecting. When you have more elevation, you have less species. And you can also keep checking on patterns. For example, you can check the temperature because temperature is important for metabolism. More temperature, more metabolism, more recycling of nutrients, and more active breathing. Can they be affecting? Another set of data across the globe and shows that the summer temperature is positively correlating with the number of species. Same in the aquatic habitat, for example, sea surface temperature. When you have more temperature, you have more species. You can also check evapotranspiration. The amount of water is vaporing through the surface of terrestrial area or water surface, it doesn't matter. And evapotranspiration, when it is high, it also tends to have more species. So that's another aspect. So maybe the temperature, energy available for metabolism is affecting this, because all these things we know that is changing with the latitudes. Mid latitudes have more temperature, mid latitudes have more nutrients and less elevation. Elevation is a complex thing, and we will come to that later. But you might notice that the bird richness was maximum in South America, and also the fish richness was maximum in South America, especially in the tropical region. So, not always, like in the Australian case, but species groups tend to co-occur together more often. We call it congruence. For example, the number of marsupial species are correlating with the number of bird species across different samplings around the globe. The number of frog species tend to correlate with marsupial species again. Number of marsupial species also correlating with snake species with less significance. So why? And this congruence could be because they are responding to the same environmental conditions. More resources means more bird species and more marsupial species, or else maybe the diversity of interacting groups of species are also promoting more diversity. It's called diversity begets diversity. How? We will come to those topics later on. Another thing that might be affecting is the stability. Stability is important because if you have frequent disturbances, cases could be the volcanic islands. Volcanic islands tend to have less diversity. Why? Every 40, 50 years, you have an eruption, wipes out the species, and you need to wait for the other species to recolonize and re-speciate to be able to increase the diversity. But how it works across the globe? There was a very nice paper by Brody, it was published in 2011, and they asked for this question. And here in this hypothesis, they checked the deglaciation, the glaciation cycles, 10,000 or 21,000 years ago, there was this last glacial maximum. The Earth was, most parts of the high latitudes were covered with ice, and it was a big disturbance. People lost their habitats. And since then, we have a more stable conditions, warmer climates. And this means that the time during this last glacial, interglacial cycle, this quaternary interglacial period, if we check the stability, does it correlate with the diversity? Because this means more stable conditions, maybe it will get more, or it will explain part of this diversity we see on Earth. And why this stability is important? One example from this paleoecology, let's say, paleology. 35 million years ago, during the Oligocene world, Oligocene to Miocene transition, the world was much, much warmer. 6 to 10 degrees warmer, very wet. And you had crocodiles in Europe, hippopotamus in England, of course not England, but the terrestrial area that represents England now, or the parrots in the temperate forests near the polar zone. No ice caps, and very warm, very tropical to subtropical environment across the globe. But now, since then, the world has been cooling. So there is change in space. And the key is that the pace of this change is important, because if it is instantaneous, and you lose all the warm areas where the crocodiles are living, you will lose the crocodiles because they have, it's instantaneous. You will lose all the habitat, they will basically freeze to death. However, it is slow enough So the crocodiles can migrate to south while the cold is coming from the north. This could be active migration, that the crocodiles are running away from the cold, or it could be more population dynamics. So every population cycle, they have off-springs, they migrate a bit more south, and step by step, they colonize the warmer environments on the southern parts. And they checked it by using these quantitative methods, by species distribution modeling. So they checked through time, how the available habitat moved away and what is the speed of this movement. And this is called climate change velocity, how the climate optimum is moving through the space. And they've seen that the climate change velocity is strongly associated with the current endemism and biodiversity. Areas with high velocities were associated with marked absence of small ranged animals. High velocity of this kind of high velocity is associated with low endemism, and the low velocity areas are basically essential refuges for Earth's many small range species. So that was a very good example how the space-time interaction through deep time and also recent time might be affecting the patterns that we see on Earth. You can see that there is a single phenomenon. We see many species at a certain location and less species in another location. And there are numerous potential explanations for that. And another potential explanation for this peak in the diversity in the mid-latitudes comes from another perspective. Not all these interactions, but much more mechanistic and much more geometric approach, which is called geometric constraints on biodiversity. It comes from Colwell. They try to understand how does the latitudinal richness patterns require any, if it's required any ecological mechanism. And they basically propose that if you have a confined space, like the Earth, terrestrial areas on Earth, it's a confined space. If you are in the middle of this range, you tend to have more species just because of niche filling in this geometric space. If you are at the corners on the edges, you can only fill these edges with small ranging species. However, in the mid parts, you can have all species with variety of ranges. So that's called mid-domain effect. So the middle part of the domain tend to be packed with more species, while on the edges, you tend to have less species. It's not about the ecology of species, it's not about nutrition, it's not about anything. It's all about the domain effect. And can it be the case? It correlates well. So that's another approach, that's more neutral approach. So you don't count in all these complex ecological interactions, trophic interactions. You don't account for the identities of species, but you only account on their apparent dynamics about mechanistic relationships like these ranges and a enclosed domain. So the center of the domain tends to be filled with more species, and the edges of the domain tends to be filled with less species just because of the geometric constraints. And it seems that they can easily account for the patterns that we see. With the optimization of this mechanistic approach, you can easily see that here you can easily replicate the same pattern that you see in nature with a model that doesn't include any information about the species, ecological identities. But that is, again, another approach. There were further approaches. One of them is from this prominent ecologist, Jared Diamond. He's also a best-selling author. And he did his PhD in the Bismarck archipelago. That's this islands located near to each other. And this is almost semi-experimental because it's a very nice setup. You have a lot of tropical small islands, and he basically sampled the birds across these islands and trying to put them in a table. And he had noticed that soon enough, some bird species never co-occur together, and some of the bird species are always occurring together, and so on and so forth. Then he noticed that he can formulate a conceptual framework which is called checkerboard distributions, where it includes forbidding combinations of species. And basically, the main understanding of it, you can understand the distribution patterns in terms of interspecific interactions. Some species co-occur, some species do not co-occur. And this brings us to another concept that is called the island biogeography. This one was an extreme case that you try to understand the community assembly with interspecific competition. This one tries to answer the same question, the biodiversity in islands, in a complete different framework. And we come back to this MacArthur that he observed the bird species across the tree, and also Edward O. Wilson, a Harvard ecologist, who is also a kind of father of the term biological diversity, and definitely a very prominent ecologist and expert. So what they together formulated was a theory that tries to explain the diversity on an island in terms of immigration from mainland and the extinction, local extinction in the island. So they try to understand if they can model how many different species on an island, basically by these two mechanisms. What they rely on? They rely on a few basic assumptions. The number of species is an equilibrium between two factors. The first factor is that the number of species coexist on an island is affected by immigration of new species from the mainland, because mainland is the long-term species source and islands are the sinks. So species are coming from the mainland to the islands. And the second big explanation is the local extinction in these islands. The extreme case is the volcanic islands. There are things happening, bad things happening in the islands. It can be an asteroid, it can be a tropical storm, or it can be another factor that results in extinction of species in the island. And this overall framework, which involves the island community, is an equilibrium state if the immigration and extinction rates are equal or kind of balanced through time. Basically, it says that the number of species you would find on an island is the interaction of species extinction curve and the immigration of new species. Wherever it crosses, you will find the number of species present on an island. And the important part here is this: how near you are to a mainland. But of course, if you are very near to the South American continent, you will receive much more species from this rich area. Because if you are near, less distance, and by chance or by basically determined flying, whatever it is, you will receive more species. And imagine that the extinction is constant between the islands. The near islands will receive much more species and end up with having a lot of more species in the island than the islands that are far away from the mainland, which is the source of species again. And check the other side of this paradigm. Imagine the immigration is the same across islands. The small islands will have much more extinction by chance, because you can have a volcano in an island, but volcano can only affect a certain distance, 10 kilometers. If you have hundreds of kilometers of large islands, the eruption of a volcano will not swipe out all species. Same for tropical storm. A tropical storms can easily erase a small island and destroy everything on it. But if you have a large island with mountains and other things in it, there is always some refugia for the species to survive. So you expect the extinction rate of the species will be higher in small islands, and thus less number of balanced species with immigration, while the larger islands can sustain lower rate of extinction, and then kind of interaction with the immigration at a point which can sustain more diversity in large islands. When you put them together, what we see, a big conceptual framework explains the biodiversity in an island in terms of immigration from the mainland and the extinction in the island, all conditioned on the size of the island and the distance to the mainland. So a small island, which is far from mainland, has the least number of species. A large island, which is very close to the mainland, which hosts a lot of species. That was fantastic conceptual understanding of these dynamics. But you can see that it is all theory up to now. But that is an elegant theory that explains lots of things with simple mechanisms. But it didn't stop here. Wilson was a kind of student of MacArthur, and this Simberloff is another prominent ecologist, was a student, PhD student of Wilson. They undertook a big experimental study to see if this theory holds on the ground. What they did, they found some islands, very straightforward. They go to the islands and check if this conceptual model works. But that's a different era for us. I mean, we are right now lots of researchers, and you are candidate researchers now, and making experiments is difficult. Here, how they do it with internal funding from Harvard University. And I don't know how they managed, but they managed to get a research permit for it. What they did, they go to the mangrove islands and they work with the ants. And they basically go to different islands and they defaunated them. They basically wrapped them around with a big cage and a nylon wrapping, and they basically put insecticide in. They killed all the ants in that island and all the other invertebrates, of course. And they monitored these islands for 2 years to see which islands have more species of ants and which islands are less species of ants, and does it concur with the expectations of this elegant framework, conceptual framework, which is island biogeography. So they basically did over 7 islands with varying size and distance to the mainland. But that's an amazing ecological research, probably not possible in this age. We will never get such a permit to defaunate an entire island, and it will be very difficult to convince reviewers now in the national funding agencies to get some funding to do this. It's amazing that it was possible to do it with university funding. That's also something very good with big research universities, and do it with that scale. The results were amazing, and it was completely in line with the expectations. The amount of ant species differed between islands at the end of 2 years, and it is definitely a function, perfect function of the distance to the mainland and the size of the island. So it also provided empirical evidence to this elegant framework, which is central to community ecology. Of course, at that time, empirical evidence was the main source, because who can go all the islands and sample them? But now, with the accumulation of research, we have lots of fauna and flora inventories across the islands of the globe. And we can map them with this high computer power and try to see the patterns. And it is the same, and you can even visually see it. For example, Madagascar is a big biodiversity hotspot. It's a big island near to Africa. All these oceanic islands, they are far from mainland, they are small, and there are so little species. So here an island hosts, these are floras, the plant species, only 1 to 120 different plants. However, these big islands near the mainlands host up to 10,000 or 15,000 plants. So the island biogeography holds very well with the global data. So you might ask, why ecology? Why it is so important to understand the biodiversity over an island? But think about island. What's an island? Is island only a land surrounded by sea? Yes, that is the kind of definition of an island in vocabulary. But in ecology, island is anything. For example, the previous lakes surrounded by terrestrial space, they are islands because they're isolated habitats, consistent habitats that's separated by a physical barrier. Forest patches among the agricultural fields, they are actually islands of communities because a butterfly living on this island of forest, a fox that is living in this island of forest, they are separated by agricultural fields. And also the mountains. The rare endemic plant or a bird living on the top of this mountain is isolated by space to the other summit of a mountain. So islands are essentially very important. And here you see the protected sites in Turkey and Anatolia. And you see that these pictures are islands of natural sites separated by urban areas. So the island biogeography can be applied anywhere. And actually, it was heavily applied for understanding communities. One of them was from Diamond for his checkerboard distributions. And he noticed that it can also be a very good framework for designing better natural reserves. So you are the Natural Management Resource Authority, and you have some Natural Management Authority, and you have some financial or political resources to decide on where to declare a natural protected zone. So then you are left with a decision. Should you have a single big patch or a one small patch? A moderate big patch or smaller patches? Or separated patches in far places or nearby patches? A square design or a line? Or a connected small patches or isolated patches? And so on and so forth. Or what about the edge effects? All these things are determined by the theory of island biogeography. So in general, a larger area is better than a small area. A connected patch is better than isolated patches. And nearby patches, even isolated, is better than far away patches. More connected area in terms of triangular design is better, a linear arrangement with more isolation from the ends. If you can have ecological corridors, it is better than isolation. And if you can decrease the edge effect, it is better than more edge effect. So this is a big chunk of conservation management relies on island biogeography. But this involves another, island biogeography was a breakthrough in ecology, because this is one of the first times it involves immigration and immigration. So communities are not stagnant, but it involves population that are exchanging individuals. That was very, very important and a big breakthrough in thinking. And you can easily reflect on this. You might remember this climate change velocity to understand the interaction between the stability and local richness. But you can also use this knowledge to understand if immigration is still in effect. And one nice paper from Jens Christian from 2007, and Jens Christian was also one of my PhD supervisors. He was a great and clever scientist. And in his work, they noticed they just worked on the forest plant species across Europe. And they used a very quantitative method, which is called species distribution modeling. What it does, you have the environmental layers. You know the humidity, you know the temperature, you know the soil type, you know many things. And these are basically essential environment for plants. So for plant species, you can model their environment. For example, for an oak tree, which are the places he can live? For example, these places is optimum for an oak species. Then you know the places that it occurs. And you can match those things. And when they did, they noticed that oak trees can live on northern areas, but they are still not there. So they are still migrating. And there is also empirical evidence for that, and also observational evidence. For example, if you take some tree species from the southern latitudes and bring them to Norway and plant them, they grow. So it is not the environment that limits them. It is the dispersal that limits them. Because since the last glacial maximum, when all these areas were covered with ice, they are extinct, and they took refuge on the southern parts. And they are still migrating since the last 10, 15,000 years to north to fill their range again, but still in action. And year by year, we can see that across Europe, the forest species and undergrowth, the shrubs and the vegetation under the forest is still migrating up north. So they are still filling their niches. So dispersal is an active mechanism to understand the community dynamics at the local space. And I told you it was only possible to understand these things because it's extremely difficult to notice them with papers of species observations. But what they do is this species distribution modeling, which is also called environmental niche modeling, where you get the species distributions, you know their ecological needs, you can use the present environment to model them. Then you estimate, by using these models, you estimate their environmental niche, and you can project it over the current landscape or a future or a past landscape. And then you can distribute their potential occurrences, distributions across these areas. All right. Let me check the time. Okay, we still have time.
Yeah, we still have 15 minutes. And
great. Sorry?
Yeah, we still have 15 minutes.
Great, great. You can stop me anytime again, and I will stop this time, and I will ask you a question. That is a fantastic conceptualization to understand local diversity in terms of immigration and extinction. And it works. And it was a great breakthrough to put immigration or population exchange or the distribution limitation to the understanding of local communities. But what is missing in this plot? We try to explain the diversity of life, trace the diversity of organisms with this framework, and what is missing in it? Any ideas? You can write to the chat. I will keep an eye on the chat. Or if you have any ideas, you can just speak up. Again, let me remind you, we are trying to understand local diversity. Exactly. Please forgive me if my pronunciation of your name is wrong, Alpida. Interaction between species, so that is good. And also there is this altitude gradient, Simeon. That's also good. So there are things that are missing. And interaction between species, so how I interact, how I can put the interaction between species or altitudinal choice. Because there is no information if we are dealing with a monkey or a tiger or a plant. The species identity is not in this framework. You don't care about whether it is plants or foxes, so on and so forth. That was also another breakthrough for empirical ecologists. So to be able to understand the community, so don't I need to know the identity of the species? And apparently without the identity of species, you can still explain the local diversity. And that also brought us to another big concept, which is called neutral theory of biodiversity. That was very controversial, but also quite a big breakthrough for understanding communities. It was written by Hubble. It basically postulates that to be able to explain a local community, you don't need the identities of species. basic diversity patterns like species richness, relative species abundance in space or time, in terms of species abundance curves or species area curves, can be explained by a framework that does not need to be involved the identity of the species. Of course, as long as we are talking about similar trophic levels, not comparing microbes with tigers. But among the high trophic levels or among the plants, we only need the assembly of the communities driven by dispersal and local extinction. And the amazing thing is that this theory works perfectly well for forest plots. It was actually, this theory drives empirical evidence from 50-hectare forest plots from Barra Colorado Islands in Panama. As I told you, so you can replicate all these species rank curves or the species area interactions by a very, very simple model that doesn't account for species identities. Of course, that's important because it provides another perspective. So there are also other dynamics might be acting without the need of the identity of species. Of course, I'm also an empirical ecologist, so it is also difficult for me to conceptualize that, I must admit. However, this involves a very important breakthrough with the island biogeography, which is the importance of dispersal. And this also led to another big breakthrough in community ecology, which makes the community ecology into meta-community ecology, where you understand a local community in terms of interacting smaller communities with immigration, emigration, and all these dispersal and spatially mediated mechanisms. And this also involves not linking one small community next to another community, but it also involves linking the small community to the wider community in the larger spatial scales through, again, this colonization dynamics and extinction and speciation dynamics. And in terms of this meta-community ecology, we tend to call 4 big paradigms, where we call them patch dynamics, species sorting, mass effects, and neutral dynamics. Basically, this gives 4 main mechanisms to understand how local factors are affecting species, which is called species sorting. In a local patch, a species, for example, this square patch, can only hold species that can live in a square patch, like A and B. But that's only one mechanism. Another mechanism is the neutral dynamics, the dispersal effects. And the others are the sink and source dynamics, like the local conditions is not optimum, but species will survive there only because it's interacting with other source populations. One example can easily be, you know that in the cities, if you stay long enough at night, you will see foxes, badgers, martens, woodpeckers. They are still living in the city. But most of the time, they are only living in the city. One example is in our campus. We have sparrowhawks, a bird of prey. And they breed every year in our campus. But every year, I'm bringing their chicks to the zoo because they always die. The crows in our campus are attacking them, breaking their wings, and so on and so forth. But every year they keep breeding. The thing is that the natural forests around us is the source population. And the chicks and the sparrows breeding them, they basically spill over to our campus and they sustain a population here, although they don't survive here. So that's a source and sink dynamic. So now we start to understand the local meta communities as a meta community connected with the neighborhoods and also with the larger spatial scales. And now we also introduced with this concept of spatial scales. And when we put all these mechanisms we have been introduced up to now into scale, we have this global species pool around the Earth, and it's basically mediated by biogeographic history like the marsupials and the plate tectonics, and also dispersal around continents or any patch. from islands to the mainland, which comes to the smaller scales where it is mediated by interaction between species or environment and species, if it is a lake or a forest. All these things, all these dynamic processes across them shapes the extant species assemblages. So communities at local scale is a product of very complex mechanisms acting over space and time across scales. Yeah, I just gave the overall, trying to put the perspective of this. So this local species, the local community is a product of biotic interactions at smaller scale, also the dispersal across scales, and then the biogeographic history, and all these things makes up a local community. Now, that's a very complex process. How you can tackle this? Definitely, this is an infinitely complex process, and it is impossible to comprehend it in the full extent. However, we can approach from different sides. And one example, seeking for empirical evidence, if a local community is related to the larger community, is coming from North American birds, they use advanced statistical models. And what they notice that if you model your local community only by the environment, you can explain 50% of your community. But if you try to explain it only by this regional species pool, if it's only a subset of regional species pool, nothing else, by dispersal, of course, you explain quite a good deal, but less than the local environment. However, if you consider both of them together, you explain the majority of the variation you see in the local environment. So it's an indirect evidence that the local community is shaped by the interaction of the local environment and the effect from the regional species pool. A similar example comes from the Palearctic phytoplankton distribution in lakes. They use thousands of Fanus Canyon phytoplankton communities, and they notice that the regional processes are shaping the local communities in relation to the local environment. Again, this relies on high-end statistical approaches and trying to find evidence support. The thing is that it is very difficult at these scales to do experiments because it's an extremely complex process. How do you experiment with the plate tectonics? How do you experiment? You've seen an experiment for island biogeography, and it's only making experiment on one aspect. So what do we do here is more like crime scene analysis and trying to find coherent support for a theory. So you make hypotheses and check for them if they support your hypothesis overall. So it's very difficult to empirical experiment on those. But overall, if we have these major factors like environment, like climate, it works at scales from plots to countries, species interactions at individual scale, individuals up to populations. The habitats as a physical resource is acting over from the local scale to the others, and then dispersal can be very active for microbes, for example, or at very larger scales like continental divides by oceans. So all these things are acting across this spatial scale. So how we see, for example, trophic interactions, how we can define them. This is an example from our own research. So when I was in Denmark, we had a fantastic data set for zooplankton and phytoplankton. And we started the analysis to understand how the local phytoplankton, zooplankton communities are forming. We were lucky because we had monthly or bi-monthly monitoring of these zooplankton and phytoplankton in 20 lakes for 20 years. That's a huge data. And phytoplankton are unicellular plants living in the water. And zooplankton are the small invertebrates that are mostly feeding on these phytoplankton. So two trophic groups, primary producers and primary consumers. And we want to see how the richness patterns are formed. there are many aspects to look at it, and we had used many aspects, and in this paper I will show you one aspect, which is we have seen this congruence. When we tend to have more phytoplankton richness, we also tend to have more zooplankton richness. This is the alpha diversity. We see the same for beta diversity. When we say more phytoplankton dissimilarity, we tend to have more zooplankton dissimilarity. So overall, for these macro diversity patterns, we see interesting thing. The diversity in the primary producers correlates with the diversity in the primary grazers. And how we can understand that? First thing could be the environment. For a good environment might be sustaining a more diverse phytoplankton community and also sustaining a more diverse zooplankton community. But of course, another approach could be dispersal. Maybe the dispersal across the space can affect positively both phytoplankton and zooplankton in a lake. Or it can be the trophic interactions, because these are two strongly coupled groups. Zooplankton are eating phytoplankton. How we can understand what is affecting what? What we did is basically, we have all the environment for all these samplings. For example, this is a lake, which is sampled for 20 years. And we know the diversity of phytoplankton and zooplankton. We know their community structure, which groups of phytoplankton were there throughout this time. We even have data on who identified what with what efficiency. And we have this data for both zooplankton and phytoplankton. And then we also have information about the environment, total phosphorus, total nitrogens, the climate, everything is there, resources, and everything is there. So what we can do easily, we can basically model the zooplankton richness with respect to all this environment. And then we can also take the phytoplankton, and we can also model this phytoplankton with the environment. But right now, I'm only giving you the zooplankton perspective. So we can model the zooplankton communities with the environment, and it only explains 12% of it. But we also put the diversity of phytoplankton in this model. We see a great increase, 50% increase in the patterns we explain in zooplankton. And we can do the same for phytoplankton. We only use the environment, then we explain to a certain level in phytoplankton. But when we put zooplankton in, we explain extra. And this is almost 50% increase in what we explain. So what we formulate in this, and we found empirical support to this hypothesis, is that phytoplankton and zooplankton communities are affecting each other beyond how they are co-affected by the environment. And this empirical support is basically a good support for our hypothesis. How they can do that is another question, because it's called diversity gets diversity. How? You imagine that you only have a single phytoplankton in a lake. That's an extreme case. That can be toxic to some of the zooplankton. So some of the zooplankton will be excluded. It can be too big for some of the zooplankton. Small zooplanktons will be excluded. So then you have less zooplankton. But when you have a diversity of phytoplankton, then there is diversity of food for zooplankton. And same thing vice versa. The diversity of zooplankton can act upon the phytoplankton, so suppress the dominant phytoplankton, for example, and then provide more relief from the competition for the less dominant phytoplankton, and then gets more diverse phytoplankton community. And overall, this is not the entire food web. The trophic interactions goes beyond the primary producers and the primary consumers. Zooplankton is a food for fish or zebra mussels. So they can be also interacting with each other, and they can create even a trophic cascade. And indeed, that's another paper that we published over this Danish zooplankton and the lake data set. And in this, what we had, again, we had data in another lake over 20 years. But this lake is special. During this monitoring, this lake experienced loss of zooplanktivorous fish. It's called, the reason is not necessary right now, but for some reason, the fish was taken out of the lake. And immediately fish removal after 1 year. Here we monitor the phytoplankton. and the phytoplankton abundance decreased dramatically. And also phytoplankton composition changed dramatically. Here you see only two colors, yellow and green. But after the phytoplankton abundance decreased, after the fish removal, we see much more diverse phytoplankton community. Lots of different groups are taking over. And we do a similar thing in the phytoplankton generations, a steady increase, and more importantly, evenness, how equally distributed these groups. We see strong increase on those. And why this is so, it is all about zooplankton. Because this fish was suppressing zooplankton community, decreasing their abundance and richness. All the big zooplankton is gone because they are more visible and eaten up by this fish. And then if you have less zooplankton, when you take the fish out, you have more zooplankton. And when there is more zooplankton, they are grazing upon the phytoplankton, getting out of this dominant phytoplankton and relieving the other phytoplankton from competition. And also all the other complex mechanisms that I explained previously might be acting to promote more diversity in phytoplankton. So we can also find support. Here, of course, there's an experiment, but it's almost like pseudo experiment in a single case. There are no replicates. But you see, we can find consistent support to a hypothesis in different perspectives. But these are kind of local. Again, this is not individual by individual interaction, but populations in the single lake or a set of lakes. But can we see the signature of biotic interactions at larger spatial scales? And there was a fantastic support for that. Gotelli is a prominent ecologist from America, with Radbeck, another prominent ecologist from Denmark. They use the bird data. Now you see the birds are quite essential in community ecology. MacArthur was looking at birds in a tree. Jared Diamond was looking at bird communities in a tropical archipelago. Sorry for my pronunciation. And I think we need a break soon. So I start to have difficulties in speaking. And then we have global bird data sets that we can check global patterns. And now people are looking at birds across Denmark. And this is only because humankind has a great affinity for birds. And for some reason, we have systematic counts of birds across the globe because of these enthusiasts and ornithologists. And this data is very valuable. And here what they checked, at a larger grid scale, 50 kilometers, 10 by 10 kilometers or 50 by 50 kilometers, they checked if the congeneric, the species that belonging to the same genera, are occurring more frequently than expected by the environment or not. And they've seen that, and of course for this they need elaborated modeling frameworks. They model the species by their environment, then they check if they are found in places that they're not expected by environment. More often, if they are co-occurring with sister species, or less often, although they are expected to be seen at a certain location, but they are not seen there. That is congeneric repulsion. So their sister species are excluding them from that area because they are competing on similar resources. And they've seen that congeneric repulsion or segregation is visible even at large scales like 10 by 10 kilometers. 10 by 10 kilometers is something that individuals cannot operate one by one. So it's population level exchange of interactions. All right. So let's stop here.
Yeah. Okay.
And I suppose this will be a lunch break.